So what happens after this histone modifications have occurred? So this is a typical process how acetylation of the H3 then causes a series of events which results in the final recruitment of the RNA polymerase to the area. They refer to it as TF2D right now because it's just the first RNA polymerase subunit that has bound and can now recruit the rest of the RNA polymerase to cause transcription. So what happens? First, you will have one or even several chromatin modifying enzymes. They will modify the histone tails. They will either remove or, in this case, add different chemical modifications. The first step here is this GCN5 hat, so histone acetyltransferase, which came in and bound the lysine 9 of the H3, so H3 is blue, remember? And attached an acyl group to this to the ninth lysine as well. It also acetylated the H4 tail, adding an acetyl group on lysine 8 in that position. Now that there's these acetyl groups found in the location, this is the binding site needed for this particular protein kinase to recognize this histone. The protein kinase cannot bind to the H3 and H4 tails unless they're acetylated. Only the acetylation in this area makes it a binding site that this particular protein kinase can recognize. Now the protein kinase has now bound, and because it's a kinase, it's another important class of proteins I hope you guys remember, it has phosphorylated the serines in position 10 of the H3. It has transferred phosphates. It took ATP and added phosphates onto the H3 tail. Because there's now phosphates on the H3 tail, a second acetylation protein will come in and now add the acetyl groups on K14s right here. Now that is highly modified histone tails now, and this histone modification sites are now what is needed for another protein complex. It's required for another protein complex binding, and that's the SY-SNF complex. That is not a chromatin modifying enzyme anymore. It will not add additional chemical modification to the histone tails. Instead, it will move the entire histone. Most chromatin remodeling complexes, such as SY-SNF, are ATP-consuming motor proteins, which will push the entire histone along the DNA. And what happens then is that this little red area of the DNA, which was previously hidden away in the inside the histone spool and inaccessible to DNA binding proteins, becomes exposed and free of histones. Now, this little area is called the Tata box, and most eukaryotic proteins have pretty decent Tata boxes. And this is a type of promoter we're going to discuss when we talk about RNA transcription. It's a type of promoter. It's a type of DNA sequence that the TF2D, so the RNA polymerase, is very eager to bind. Once the Tata box is free of the histone spool and can be easily bound by the RNA polymerase, transcription and mRNA production in that area can occur. So, two types of protein classes work with histones. There's the chromatin modifying enzymes. They modify the tails, but they don't move the nucleosomes. And it's the chromatin remodeling machines that then move the nucleosomes around, exposing promoters, for example. Now, if you want to maintain the transcription of a particular gene, do you need to go through the same process over and over again, constantly moving around nucleosomes? No. Once a particular area has been designated for transcription, other proteins can come in and stabilize the chromatin structure. So see, we just saw right now how methylation exposes, loosens up the histones and exposes a promoter so the RNA polymerase can transcribe from there. Bromodomain proteins can then also bind acetyl, also recognize acetylation sites as their binding sites, so they will bind to the acetylation site and to each other and create this very stable polymeric protein structure, very rigid, keeping this particular stretch of DNA in the open state. Similarly, if the tails of a certain histone tail has been methylated and all of the histones in the same area have the same modification tech, remember this will not work if they all have different histones have different methylations, then the chromodomain proteins will come and recognize the methyl groups and also create this polymer, rigid polymer, keeping that area of DNA highly condensed and inaccessible for transcription machinery. And again, in this case, these guys listed here who convert the tails between acetylated state and a methylated state are the chromatin modifying enzymes. They modify the histone tails, while the chromatin remodeling complexes such as the bromodomain proteins or the chromodomain proteins then create rigid structures and reposition nucleosomes, or in this case, keep the nucleosomes in a very rigid particular state, stabilizing the effects that the chromatin modifying enzymes have caused in this area, keeping this promoter always exposed and always accessible to the RNA polymerase. So the different histone modification are important in many biological processes and also many diseases. One example that I would like to use is the herpes virus. So you guys know how herpes is this annoying little disease and that creates cold sores. However, I'm not sure if you guys appreciate the fact that herpes actually doesn't reside in our lips. So treating the area here will not actually kill all of the virus. Instead, it lives in the sensory neurons in the brain that protrude into our lip-mouth area. And the herpes simplex virus has a particular protein called VP16 that can acetylate histone tails, making sure that the viral DNA is expressed. So what happens inside one's mouth is when one has a cold sore, the virus has injected its DNA into the nucleus, and it's just DNA. The cell doesn't know any better, so there's always free histones in the nucleus, and they will bind and spool up the DNA. Now the next step for any for the cell is to probably methylate it, and unless it has a signal to express it, it will not. However, there's VP16 here, which is a viral protein, and VP16 acetylates the K9 lysine and also methylates the K4, allowing for open DNA structure and for production of the viral proteins from the viral genome. That happens in the mucosal, in the epithelium of the mouth of the mucosa, because there the viral protein VP16 can get into the nucleus. So we have open chromatin of the viral genome and active viral gene expression, acute infection. That's what a cold sore is. It's an acute herpes infection. The cells of the tissue affected by the cold sore are actively in the process of lysing and releasing lots of viral particles. Now, the virus also can infect sensory neurons. And there, the situation is different. VP16 does not have the correct nuclear localization tag to enter the nucleus of a sensory protein because the nuclear pores inside neurons are slightly different. So even though the genome is injected inside the nucleus, the VP16 is not there to assure open chromatin. And the cell's histones come and they bind the DNA and create these nucleosome structures. And then, since there's no VP16 to cause acetylation of K9, K9 is methylated, which keeps the genome, the viral genome, closed, in close chromatin conformation, and no viral genes are expressed. So the infection goes latent or dormant inside sensory neurons. Now, this cell, this is not, unfortunately, just because the DNA is dormant doesn't mean that whenever our immune system is not repressing some of the virus that sometimes is made, it will not cause an acute infection resulting in a cold sore. But it's a very clever way for the herpes virus to actually persist in the sensory neurons, because it doesn't kill them. It just lives in them and periodically affects other cells. So latent infections promote the eventual transmission of the herpes virus months or years later to another host. So today we talked about how there's 4 different standard histones, H2A, H2B, H3, and H4, and another modifier called H1 that is not part of the nucleosome. However, I hope you guys can appreciate another fact, that there's other histone varieties, other histone genes that are used in particular special situations during a cell's life. For example, H3.3 is a special histone that can replace H3 and keep a particular region of the DNA in the locked open conformation, similarly how the bromoproteins were doing it. While SENP-A is another version of the H3 histone, which has these particular kinetochore attachment sites and is plugged into the nucleosomes right before mitosis, because it will allow for kinetochore attachment to the mitotic chromosomes and correct division of 2 sister chromatids into 2 different cells. H2AX is a version of the histone 2A. It is replaced in the areas of the histones of the DNA where there's double-stranded breaks, and the phosphorylation in those areas attracts repair proteins that will either use homologous recombination or non-homologous end joining to seal this double-strand break. Well, we just talked about DNA replication, and that seemed like a very sophisticated process that would likely be hampered by the presence of these big nucleosomes in the way. So what happens? Does the replisome displace the histones as it replicates the DNA? But if it does, wouldn't it erase all the important information that the histone tails carry, telling whether that particular area of the chromosome is euchromatin, heterochromatin, to be actively transcribed, to be silenced, etc. And so how can you move a histone out of the way when you replicate DNA? And what do you do? And how do you avoid losing all the important information if you do remove the nucleosomes from the DNA? So that's done in a very creative way by the replication machinery. The histones, the histone inside a nucleosome is not one solid thing. It's made up of 8 different proteins. Now, when DNA replication occurs, the replication fork displaces H2A and H2B, but it doesn't displace H3 and H4 that can move out of the way, as the H3 and 4 can bind any side of those double loop of the DNA. So as the replication fork comes through, they can be shoved out of the way. And so these marked H3, H4 subunits remain on the chromatin and are randomly segregated either to the newly synthesized strand of the DNA or the old one. sorry, into the either the lagging or the leading strand. And then as the DNA is replicated, a lot of new DNA, a lot of new histones are being produced. And these then find these marked H3H4s and bind up with them, as well as the H2As and the H2As and H2Bs come in, and then the H3s and H4s to make full octameric nucleosomes again. But as you can see here, now the histone modification profile of this chunk of DNA has changed. Some of it carries the modifications, like these light green, pale ones, and some, the bright colored ones, have no modifications because they were freshly made. So how is then the memory preserved? Well, as soon as DNA is replicated, another type of the histone modification or chromatin modification machinery comes in. And it uses the tags found on some of the histones in the area, such as this acetylation tag on H3, to then put the same kind of tag on the adjacent histone. So the bromodomain hats do exactly that. They bind. They recognize that this is an acetylated histone because that's the only thing they can bind to, so they bind acetylated H3. And because they're long enough to reach over to the next nucleosome over, they can then modify that tail to an H3K9 state as well, for example. So that is how the histone modifications are propagated post-replication, and the epigenetic profile of the different areas of the chromatin is maintained, which is really important. So newly formed and unmodified histone complexes take on the characteristics of their neighbors. Similarly, heterochromatin some of euchromatin can be converted into heterochromatin, and heterochromatin areas can expand, which sometimes plays a role in disease. Your book discusses it in far greater detail. So because the heterochromatin machinery also can reach over to the next histones, it can then place a tag and then bind that new tagged histone and place another tag and start compacting open euchromatin around the heterochromatin domain. So how do we know all of this? The technology for today is a chromatin immunoprecipitation and sequencing approach. Its shorthand is called ChIP-seq. So CH in ChIP stands for chromatin. IP or IP is the general abbreviation for a commonly used experiment type in molecular biology called immunoprecipitation, where you use an antibody to separate something from everything else. And then seq is sequencing that occurs later on. So let me walk you through the process. Let's pretend that we have this genomic DNA spooled onto nucleosomes with some linker DNA in between. And we want to know about what parts of our giant genome are carry histones with the H3, say H3K9 modification. So we have isolated histone tails from histone 3 with the K9 acetylation modification, and we have raised antibodies against them, either by injecting them into rabbits or doing it in cell culture. So we have these antibodies that will only bind the histones that have the H3K9 acetylation modification. So we take the DNA from the cell or tissue of our interest, we digest it with nucleases that will remove the linker regions and only leave the nucleases, which contain about 147 up to 200 base pairs wound to onto the nucleosome. Then we incubate all of these different nucleosomes with the antibody, and only a few of them will bind. Only the ones that have the H3K9 acetylation will bind to the antibodies, and we will wash the rest of them away. And how come we don't wash the antibodies? Well, this picture doesn't show it, but your antibodies are attached to something like a column. So everything that does not bind to the antibody is gonna go through the column, and these guys remain on the column. Then we use a clever trick to digest the antibody, which is protein, as well as the histone part of the nucleosome, which is also protein. So we'll use some sort of proteases to get rid of them. Proteases will destroy protein but not DNA, because DNA doesn't have the same bonds in them. It has a phosphodiester bond, and instead of a peptide bond. And then we purify these DNA molecules, about 200 base pairs each, and now we can sequence them. So usually at this point we don't use, we will not use typical Sanger sequencing, but because there's so much data, because you're dealing with a typical enormous genome of, say, a human, we will then do, one would then do the Illumina type of sequencing, or pyrophosphate sequencing, all the other different high throughput sequencing approaches described in the DNA technologies chapter that we covered. And we will then get the sequence. So it's sort of a joke by the authors that they showed this really ancient DNA gel, because that's not usually how things were going to be done. Most people who do ChIP-seq right now will absolutely use Illumina sequencing. And then we'll get, we will use alignment software to place whatever sequence we have gotten from these blue strands onto the human genome, if you were dealing with humans, and see if there's an enrichment of certain areas. So each single sequence that we got will give us a little peak, and the more sequences of a particular area that we isolated, that we know were bound to the histone that was acetylated at H3K9 at that moment, the larger the peak is. And one of the ways you can look at some of this information is using the Human Genome Browser from Santa Cruz. I already described you guys how to do it. So if you go to the University of California, Santa Cruz Human Genome Browser, just Google it, and put any gene, you will see the, say, the H3K27 acetylation profile in that area. That was probably one of the more massive experiments done recently. And you guys can see here that these peaks, and each peak is a single read, single sequence that was read and matched to that particular location of the genome. The higher the peak, the more reads. You can see that they're slightly different colors. That's because they actually looked at lots of different human cell lines when they did this experiment. So each color corresponds to a particular human cell line. If you actually go to the link and click on one of these peaks, you will get the color code translation as to which cell line is cyan and which one is orange and which was blue. But let's zoom in. So if we looked at this particular location, you can see that there is beginning of a protein here. RAD18 is the name of the protein. It's the beginning of the DNA coding for the RAD18 gene. And here you can see the nucleosomes, which H3K27 acetylation mark were found here in many, many, many different cell lines. And that's a very typical thing, very, very typical thing to find for nucleosomes around promoters, because that's precisely what this area is. There's a promoter in here that allows for the transcription of this RAD18 gene. So the ChIP-seq data analyzed can give a lot of information about how different proteins are expressed, just looking at the pattern of nucleosome organization. So you see here that this is a location this is the positioning of the different nucleosomes on the DNA as the RNA polymerase moves through and makes RNA. Hopefully we'll get to that subject next week. You can see that when the RNA polymerase is actively on top of the two nucleosomes, these particular locations where the RNA is being made are opened up and stretched out. And then the promoter region is also definitely opened up. The little binding site that the RNA polymerase recognized, called the Tata box, is exposed and not wrapped onto a histone, so that other RNA polymerases can come in and do the same thing this guy is doing. So you guys can see how the nucleosome organization inside close to the active promoter region has a very, very particular pattern, and then is a little bit more shifted around as the RNA polymerase goes through the gene to express. So here I would like to leave you guys with a few things to think about and derive on your own. You're welcome to email me you guys' answer. This is not for points, but you're for general growth as a scientist, as a future scientist. So think about it. Why is this particular patterning relevant for gene expression? And what could a molecular biologist learn with an antibody that recognizes a specific activating histone modification? What would be the correlation that we can devise? Now, I like giving you guys additional information for studying if something that I covered in this chapter wasn't clear. So this particular video, it's from YouTube. Here's the address. Is the chromatin and histones review video. It reviews what I talked about in far less detail. It's meant for a lay audience, but what I really like about this video is that this is actual simulations that were run using big computer clusters to show how a DNA molecule and how DNA molecule behaves inside nucleoplasm, which is water, how it interacts with all these water molecules that surround it, how mobile all these histone tails and phosphodiester bonds of a DNA molecule actually are. So hopefully you guys will enjoy watching it. And then finally, this is a summary slide you guys can read, and hopefully it will help you study about all the important points that I talked about in this lecture. So with that one, I will conclude this recording.